Well, thank you so much for the introduction. You all can hear me? Okay, I'm not that loud. Okay. So yeah, glad to be here. I really want to thank our MILLIPLEX team, Mike and David for inviting me. They help us so much. We work with them all the time. What I will do today is that I will just give you an overview of the kind of the work we do and how we incorporate Luminex platform and the MILLIPLEX kit to get more information about the work we do. So as you can see from the title, my lab works on neurotropic viral diseases. So that means the viruses that infects your brain, right? So as you can imagine, it's usually very difficult to diagnose neurotropic viral infections and also very difficult to prognosis and also, of course, the therapeutic. A lot of reasons for that. These infections are very different from the normal infections that you usually have. For example, a lot of time when patients come to the hospital and then they have all those neurological symptoms, there's absolutely no virus to be found in the blood or anywhere else. And I will show you some of the examples of that too. And even sometimes it's very difficult to find any virus in the brain as well, even if you're able to obtain the samples like CSF or something. So a lot of time it's very difficult to correlate with the pathogens, and there are many pathogens which can go in your brain and cause encephalitis. So a lot of time, the main goal of my lab, of course, is to look for biomarkers, either they are diagnostic marker or they are prognostic markers, and of course, eventually to hopefully to find the mechanism or therapeutic markers and how we can use all this information to better manage these infections, right? Other thing I also want to mention, of course, during COVID-19 is not necessarily a neurotropic virus, but we have seen cases of long COVID and brain fog and many other things with the COVID-19 as well. So I will show you some data regarding that as well. That's my title. So this is the outline. Usually when I give the presentation, my outline is quite different because I'm mostly talking about the data, but since it's a Luminex events, it's a technology, so I thought it will be good to not only just to show you the data, but also to compare the different platforms that we used. So Luminex and MILLIPLEX is one of the tools, but we also use, of course, a lot of other tools like RNA-seq, NanoString, and other platforms, and how all these data compare to each other and how we can use all these tools to, of course, to get more information. So, of course, I'll talk about the Luminex assay in my lab. I'll use an example of SARS-CoV-2 infection to study neuroinflammation and compare two different platforms, NanoString and Luminex. I'll show you other advantage of using these kits. It's one of the vaccines that we have worked at that's actually in the clinical trials and actually also in some of the patients as well in some countries. We'll show you some of the data that we obtained and how looking at immune response at a broader level can give you a lot of hints how vaccines are working. And then I will move on and show you one example of validation of RNA-seq data using these kits. I'll also show you one example of how we can adapt and use these kits for different species. And also at the last touch upon some of the assays that we have developed using this Luminex-based platform for the diagnostics of different pathogens. Okay. So my lab, we are a few miles away from here at Georgia State, downtown Atlanta. So if you don't know, we have a beautiful animal biosafety level 3 or biosafety level 4 facilities. So we are able to work on many different emerging pathogens. We have huge facilities, so we can use many different kind of animal models. So of course, we use mouse, but we also use hamsters. We can also use ferrets. Most data I will show you today will be of the mouse. So we work on different viruses. SARS-CoV-2, of course, is pandemic. So we have worked a lot in the last 3 to 4 years on SARS. I'll show you the data. But most focus of my work and what I will show you is mainly the encephalitic viruses that are naturally go and infect your brain. Some examples are West Nile, it's a mosquito-borne, Zika virus, Powassan. I'll show you some data about Powassan. It's a new tick-borne neurotropic virus. Then we have Japanese encephalitis, Usuttu, and many others. So we have animal models for all these. We have nice longitudinal models. So we study them. We see what happened before the acute encephalitis, what happened during the acute encephalitis, what happened when the animals recover from the acute encephalitis. So a lot of data can be generating using all those models. Of course, this is Luminex event, so I thought I should focus more on like the Luminex assay. Of course, we know, it's a very good system based on we can multiplex things, and that's very important for us because we usually have a very small sample. We do mouse study. If we have to do ELISA and other things, it's very difficult for us. Using a very small amount of sample can look at many analytes, give us a very nice overview of what's going on in the animal. And of course, we can do different kind of assay. These are not only amino assay, there can be nucleic acid assay and enzymatic assay. And of course, you can also have assays for specific detection of pathogens, and I will show you one of the example for using the West Nile. We mainly use this millipore, MILLIPLEX kit. That's how we are so close to millipore people. But we also have developed our own assay using this platform as well. And we use different kind of sample. Most of our samples are also tricky because, they're very viscous. For example, CSF is very difficult, usually very small and very thick. You can have many fatty tissue, like brain is usually a fatty tissue. It's a little bit difficult compared to if you look at serum. So, and like fat tissue. So we have been doing this assay for a long time. So we have done on various different kind of tissue. I won't be able to show you all the data, but I will focus primarily on the brain, okay? So first thing I will discuss is using this example of SARS, how we were actually the very first lab to actually show in an animal model that this virus is primarily infecting the brain after the lungs. So we did a lot of studies to evaluate what's going on in the brain of these mice, and of course we did this large genomic data too. But this example I want to show is with the nanostring. So nanostring is another platform, encounter platform, right? Sometimes what happened with the RNA-seq, you're amplifying the things, and sometimes they don't correlate with what's actually happening. But with the nanostring, it's like a digital platform, so you're not amplifying anything. You are actually picking up the actual copies of RNA. So we found it actually much more accurate. So this has a different platform, of course, looking at RNA, not the protein. But I'll show you how good the data match when you look at the RNA level with the nanostring, and then you look at the protein level with the Luminex, and how it's interesting to identify very specific markers so that they can correlate with what's going on in the animal. So to show you, before I show you the data, of course, I want to explain you the model, right? So for the COVID-19, of course, we grow different kind of brain cell, we grow organoids, we grow primary cells, so we do a lot of these cell culture work and infection, and then look at different response. We also work a lot on animals, so mostly mouse. So we use this what we call humanized mouse. These mouse express the receptor, human receptor for ACE2. It's a very lethal model, so I'll show you the data that using this model and how we were able to evaluate neuroinflammation. So this is a busy slide, but what it's showing is that it's showing how this model works. So basically, you have these mice, they express human ACE2. Normal mice would not get infected. So like I said, my lab was one of the first one to show this. So we use this mouse model, and you can infect them with different variants. You can see these are B.1 is Wuhan, then you have UK, South African, Delta, mouse adapted Omicron. So we use all these different variants. You can actually see very nice differential pathogenesis of these variants in these mice as well. So the point is that when you infect with these SARS, these animals get really sick, right? Almost all the animals die. But you can also see a difference between, for example, Omicron versus South African or Delta. So they are more pathogenic than the Omicron. So you can, and that's what we see in the humans as well. So you can see that kind of difference in this mouse model as well, right? You see beautiful infection of lungs. Almost every cell in lung is infected. It's a brilliant infection. So this is a slide showing the, a cross-section of lung of one of these mice. We stain them with the N protein, that's a SARS nucleocapsid protein, and green is double-strand RNA, that's the genomic RNA that made during the replication. So as you can see a lot of red, that's a protein, a lot of virus, a lot of viral protein. You see a lot of green as well, that's basically showing all the active virus replication, right? But so this is very common to see in these models. But more than lung infection, the thing with this model is that eventually the virus goes in the brain, and then it causes what we call encephalitis, right? So you can see a lot of virus in the brain as well. A lot of neurons are infected, right? And then of course, you see a lot of pathology as well. In the brain, this is the H&E staining showing, some of the infiltration of the cell as well as some dead, I don't have a pointer, but believe me, there are some dead neurons in there too. So anyway, so since we saw this and we work on neurotropic viruses, so we thought like we want to study more, want to see what's going on in the brain, right? So of course, first thing we did was to look at these brains at different time points. You see this one is showing the viral load at different time points. So at day 1, when you infect the mice, you don't see any virus in the brain. Day 3, you see a little bit. Day 6, when the animals are actually dying, you see a lot of virus in the brain. So that's kind of a correlation that, at least in this model, this brain infection is killing these animals. So in this study, this is the viral data. And then on the other side, this is nanostring data. So we took this brain, extracted the RNA, did this nanostring assay. So, there are almost 700-800 genes. I'm just showing you some selective genes here. So as you can see here, this is like day 1, day 3, and day 6. You see at day 1, you see nothing. There wasn't any virus in the brain. Day 3 also, you don't see a lot. There is some faint. It's difficult to see, but there's a little bit here and there. But at day 6, you can see a lot of, these are highly upregulated, so that goes hand in hand with the infection. But you can see there's a lot of differential. You see there is CXCL10 and CCL4 will be very high, while other ones are, even though IL-1 alpha, beta, which are very well known, wasn't that high in the brain, right? There's one important thing I want to mention, when we talk about immune response in the brain, is very different. A lot of our knowledge comes from immune response in the periphery. We study macrophage, we study T cell, we study B cell, we study sphenocytes. But how these cytokines are produced in the brain and from the brain resident CNS cells are quite different. Their levels are quite different, their basal levels are quite different. So we usually see a very distinct response between the brain cells or between the peripheral cells like macrophage. But anyway, so this was a good data to collect because this gives us selective markers, selective genes that we can further look into to see if they are, what kind of role they are playing. But for the purpose of this presentation, of course, we wanted to look at the protein level, right? So this is the same samples, exactly same samples which we use for the nanostring. So this is the Luminex, using the Luminex platform, the MILLIPLEX kit. So you can see here, of course, there's a lot of bars here, but the main take-home message, you can see the day control and day 3, you barely see anything. They're all very basal level, the same thing what we saw with the nanostring, right? But look at most of these are highly up at day 6, the same that we saw with the nanostring. And actually, it correlates very well too. Only the one that we saw with the nanostring that you see high here as well. And even level of upregulation is also very good. So it was helpful for us. So we did a lot of other studies, but I want to show you some other examples. So this is just one of the examples that I was showing you with the COVID-19. This is another example with the vaccine. So it's a MVA-based vaccine. MVA is a vector, very commonly used. A lot of people, the vaccines that we normally use right now, mRNA vaccines, they mostly, protection is based on antibodies, right? Not necessarily T-cell response, but there are a lot of immunocompromised people who cannot make that much level of antibodies, or older people who cannot develop that kind of antibody response, so T-cell response becomes very important, and vectors like MVA are known to induce a very substantial T-cell response. So I'm not going to do too much thing. The main thing I want to show you is that, if you use this vaccine, even if you use single dose, or of course, if you use 2 doses, all the animals are protected. Of course, if you don't have vaccine, all the animals die. And another important thing here is that even those animals who are all protected with 1 dose, they have no neutralizing antibodies, but still they are protected. So you can have a full protection in the absence of neutralizing antibody. But other important thing was also is when you look at the cytokines and chemokines. So these are from the lung homogenates using, again, the MILLIPLEX kit. Again, lot of data, lot of points. But the main message is that most of the cytokines are up if there is no vaccine. So if you have virus replication, cytokines go up. But some specific cytokines like RANTES and others, which even in the absence of infection, the vaccine is inducing that. So there are some differences, even though all these cytokines look similar, but the role they play can be very different. So RENT is a very important for T cell differentiation, for T cells. So that's what this vaccine is primarily generating. So again, just one example of using all these assays to make sense of the data and also to gather more information about the specific biomarkers that may be useful. Okay, so I still have time. So I'll move on and show you another example. So this is basically, we do a lot of RNA-seq. I told you, right, we have these models that we use, we get different kind of tissue. So usually we want to validate this data at the protein level, so we use, of course, this platform and these kits all the time to validate our data. I'll show you one example, so see how good it looks, right? So the example that I will give you is about the Powassan virus. Most of you haven't heard of, but it's a emerging virus that's growing. It's a tick-borne. So ticks don't fly, so we don't see a lot of cases of Powassan, but wherever you find ticks, you see cases of Powassan. It's a very lethal infection. Virus goes in the brain. Most people get acute encephalitis, and even if the people who survive, they have a long-term neurological sequelae, right? So we have in our lab is a very nice mouse model. So this model behave very similar to what happens in humans. Like you get infected, and then initially, in a minute I'll show you that. So this one is a example of these are regular black 6 mice, and you can give a very tiny bit of virus on the footpad, like what would happen in a mosquito bite, a tiny bite, and the virus will take a week or 8 days to get into your brain, in the mouse brain, and same thing happens in the human as well, and you get the encephalitis, right? So how we have made the model is that we have figured out a dose where we can kill 50–60% of the animal, but remaining animals survive, and we can study them, long term. And it's actually a very interesting model because, initially when you look at virus in the blood, you see a very high virus in first 2–3 days, but there's no symptom, no weight loss, nothing. But by the time it's day 6, almost all the virus is gone from the blood. So you can't find a virus. But at day 6, virus starts to show up in the brain. And then it goes even higher at day 8 or day 10. And that's the time you see all the symptoms and the animal die. And that's what happened in humans. So at that time, when that animal has died, you cannot find any virus or any antibodies in the serum. And sometimes, even if you go later time points, these animals keep on showing all those symptoms. And they have very small amount of virus, which is usually hard to detect, but can only be detected in the brain. And you get the brain tissue. So you can imagine what kind of work we try to do. We try to see if there are some correlation, if we look at all these biomarkers in the blood and keep looking them in the longitudinal model and see what's the outcome in the mouse as well, and then also look at the biomarkers in the brain at the pre-acute, acute, and the chronic kind of stage, and see if we can come up with the algorithm that can show either diagnostic marker or prognostic markers or things like that, right? This is also a very good model for blood-brain barrier leakage, So this is another thing that we don't know much about. So these viruses, they open the barrier. It correlates very well. When the virus is only in the blood, the barrier is tight. Virus starts to go in the brain, little bit leaky. There's a lot of virus in the brain, the whole barrier is leaky. And then the virus comes down, the barrier comes back too, right? So all those things can easily be mimicked in this kind of model, right? This is just showing some slides from these animals showing that there's a lot of virus in the brain. The virus replicates in the brain like so much. Almost every neuron can be found to be infected. A lot of neuron death happens, a lot of pathology, a lot of immune cells going from the periphery into the brain, right? So it's a very, very good model, very, very severe model. Anyway, so in this model, what we did, of course, to look at the, data over the time, we killed these animals and we looked, day 3 when there's very little virus, day 6 and 8 where there's very high virus, and then day 21 when again there's very little virus in the surviving animals. And then we could take out the brains and we can do the RNA-seq, right? So this is an overview of the data. You can see a lot of genes that go up, a lot of genes which are down. But there's the same pattern. At day 3, you don't see much. 6 and 8, there's a very high upregulation. At day 21, there's not so much. But there are some specific things you can still keep seeing at day 21 as well. So there are few things show up very early and keeps there till day 21. Few things are only during the acute encephalitis. So all those things you can, and not only the upregulation, you can look at downregulation as well. For example, this T cell response, as the days keep going on, the T cell response actually keeps going down. So this is something that the virus does, right? This is the RNA-seq data again, but basically all the cytokines and chemokines, because that's what we use the Luminex platform to validate as well. So you can see these are the mRNA level from RNA-seq. So you don't see at day 3, but you see at 6 and 8. You can verify, usually people can do a RT-PCR. It's basically the same thing, but you can look at the individual gene just to validate some of the data. So there's a good correlation between RT-PCR and RNA-seq. But of course, when we do Luminex, so this is done with the MILLIPLEX kits. So you can see these all, see the same data. You don't see anything in the control. You don't see much in the day 3. You see very high at the day 6 and day 8. And there's a differential kind of expression. Some of them are very high. Some of them are barely. These are all the cytokines. Very similar things about the chemokines. But in general, if you correlate with the RNA-seq data, it correlates really well. So the genes that you see very high there, you see very high here as well. So, so again, it's one example of using this to gather more data, to validate our data, and how we can look at long-term effects, right? So I will give you another example. Again, they're random things, but we do a lot of stuff. So this one is some time ago. I don't know if you heard of, there was an outbreak of a Zika virus. So Zika virus is something that could infect pregnant women and go to the unborn child and could go to microcephaly, right? We were actually the first patient in the US was actually diagnosed by us in the Christmas 2015. So a woman came back from Brazil and gave a birth to a microcephaly child. So and we got the sample and we isolated Zika virus from that. So, but anyway, but our work is mostly not with the human patient. We work with the animal model. So our goal was always to, the virus came up, we need to make an animal model. But there was an issue with the Zika virus that, unlike the data I showed for Powassan and West Nile, Zika virus doesn't replicate in mouse. So you need to have a immunodeficient kind of a mouse to get the Zika virus replication, which is usually not ideal when you are studying the immune response. So what we came up with a new model for the guinea pigs. So guinea pigs were actually for studying reproductive or studying pregnancy-related thing. Guinea pigs are actually much better because all the pups are fully developed when they're born, unlike what happened in the mouse. The gestation period is so small. You can't study all the trimester and things like that. So we were the first one to make that model, and we showed that, and many other people after us showed that this model actually works much better for Zika than the other viruses. So this is some data showing that when you infect pregnant guinea pigs or regular guinea pigs, you can detect the Zika virus or NS1 protein, that's the viral protein, in the serum by PCR and ELISA as well, right? But we also wanted to look at the immune response, right? And the problem with a lot of these models is that they don't have kits available, so there's no kit available to look at cytokines or chemokines in the guinea pigs, right? So we talked to Millipore people, and they recommended us that we could try rat kit, which actually have shown to have cross-reactivity with the guinea pigs, not for all the analytes, but many of them. And we were actually able to see that. And after us, other people also used that as well. So this is just some data showing from this serum of these guinea pigs, blood collected from these infected guinea pigs, and then use the rat kit to detect the analytes in the guinea pigs, which actually worked out really well for us, gave us some important biomarkers and targets to look at. So again, another example. I'm almost at the end. I'll give you one more example. So this is something also we were doing a lot a few years ago. We haven't, right now we're not doing that much because we got so much busy with the COVID. But this is another focus of the lab. So we look for diagnostic assays, So of course, antibodies-based diagnostic, IgG, IgM, are an important serological assay for detection as well. The regular kits that we have, ELISA or other, again, they take longer time, more samples and things like that, and we work on mouse, so our samples are always limited. So we use this LumenX platform. We coated the beads by ourselves with the viral protein, and then we could use the samples to test that, well, this assay work as good as if you would use the traditional ELISA. And this can be multiplex. So with Vaginal, we could add GCA, we could add Povastin, and we could do a multiplex immunoassay to detect all those things. This is just one example of using the Vaginal. This is another example of using the same assay for our subsequent studies, because we were using mouse model, but then we want to show that how the antibodies are going up or down, and this assay, and other people have also used this assay as well. So with that, I will conclude. A lot of things I told, I don't know, hopefully some of them were helpful. But we like these assays, so we find very comparable results to ELISA, NanoString, RNA-seq, or other platform. It's a good way of reading them using this, cytometer based. It's very rapid. It's more sensitive, more specific. Most important thing for us, require very less specimen volume. Another important thing, we can multiplex and look at the broader picture, and we can also develop the serological diagnosis of viral diseases as well. Okay. So with that, I want to thank all the members of my lab. Some of them are sitting right at the end. So thank you, and I also want to thank all the funding, and of course, LumenX and MILLIPLEX for having me. And I'll take any questions you have.